My name is Raj Agrawal. I'm the Vice President for Clinical Development and Therapeutic Area Lead at Ray Therapeutics, which is a small biotech company in Berkeley, California. We also have a presence in Irvine, California, where we have a testing facility for endpoints and other aspects that we deal with in the clinical trial. I'm an ophthalmologist and a retina specialist by training and experience in clinical care for many years before I took a jump, so to speak, over to the other side and started working with industry. This is my first time attending the Choroideremia Research Foundation's conference, and I've had an excellent experience in, first of all, meeting a lot of patients and patients' families, getting to know some of them in some more detail, and also meeting colleagues in the space. It's, this kind of experiences really inspire somebody who works in this space in terms of trying to make a difference, to keep pushing ourselves more and more to try and make a difference and get some treatment for patients. So it has been really an amazing time the last few days here in this beautiful city of Minnesota, and to spend time with the families and members of the foundation. So one of the things I was really amazed by is the fact that the families and the patients themselves are so very aware of what is going on in the space. So they're very well informed, and not only are they informed, but they actually are coming up with ideas and suggestions as to what we can do to make the space and the field go forwards. I met some of the scientific advisory board members in meetings, in the conversations. Obviously, some of them are patients themselves. And to hear from them as to what they've been trying to do for the last so many years and decades, and in helping clinical studies, trying to get us to the next level, and also in conversations with the regulatory agencies to try and see whether we can push in terms of endpoints and other suggestions that can help potential approval for therapies. I'm really amazed by that. Besides that, so some of the family members did express, that they've been involved in some other programs before in terms of being part of the clinical trials. So they wanted to know how do they get involved in subsequent studies. So there were conversations about that as well with the families and the patients themselves. Obviously, people are keen from patients' perspective and the family perspective. They are keen to get some treatment that can potentially help either stabilize the disease or improve the situation in some ways. And to know and to become aware of that was inspiring in a way for me. So, like I said again, it was a great time talking to patients and the families here. So we, Ray Therapeutics, we have focused on optogenetics. And optogenetic space is relatively new. It's a type of gene therapy. But the idea behind optogenetics is we are trying to recreate the functionality of rods and cones, which are the photoreceptor cells, in the cells that are a part of the visual axis, but they are not really light sensitive themselves. So what we do is we inject light sensitive proteins in the eye through an intravitreal injection, and that transduces the cells like retinal ganglion cells, which form the pathway to the brain, but they don't really get stimulated by light. So by converting them into light-sensitive cells, so when the light falls on the retina, these cells now get stimulated and they create a visual response, which then goes to the brain, and that's how the patient is able to see again. Obviously, early days, we have started a clinical study. We already dosed one patient, but we have enough data from preclinical work with animal work to be suggestive of the fact that things will really work well in patients as well. So the animal data that we have so far suggests that there is significant improvement in visual acuity in those animals. Those are animal models, which is called a TKO model, which allows us to kind of see what can potentially happen in a patient once we do the treatment in patients. So what we've seen in animal data is that there is significant amount of improvement in visual acuity in those patients, in those animals, but there's also improvement almost to normalcy with contrast sensitivity and also mobility, and which are all important issues in terms of patients' day-to-day living. So we expect, again, this is expectation from animal data to human outcomes, but we expect something similar to happen in human patients at this time. So like I said earlier, we do have an ongoing study at this time. Obviously, this is a phase 1 study, phase 1-2 study in a way. So what we have focused on majorly is safety in patients. Obviously, first patient got injected recently, so we want to make sure that what we are dosing in patients is safe enough, and obviously we'll also be evaluating those patients for outcomes in terms of visual acuity and mobility and things of that nature. So actually, this is good because that message can get out to patients, right? So the clinical study currently focuses on patients, obviously, with proven choroideremia. So we do ask for genetic testing to be done on patients so that we know the patient is choroideremia patient, number one. Number two, what we're looking for is, because again, this is a safety study, so our interest is patients with bad, if I may use the term, bad visual acuity, not the good side of visual acuity. So we're looking at patients who are worse than 20 to 100 in terms of visual acuity. What we're also looking for, again, because this is early days in the study, we want to make sure that we don't confuse the data that we receive. So if any patient has already received gene therapy study, was part of a gene therapy trial previously, unfortunately, we cannot take those patients into the study. In a way, we want a patient with no exposure to any genetic material previously. So obviously, choroideremia patient, 2,200 to 28,000 vision. Obviously, in terms of the rest of the list of inclusion exclusion criteria pretty much remain the same as in any other study. We want to make sure the patient does not have any ocular disease, other ocular diseases that may preclude us from examining the patient, us meaning the sites, from examining the patient and trying to find out what has happened in the patient post-injection. So those are the criteria. In regards to the clinical design, trial design, we're looking at 2 cohorts of patients, and each cohort is 3 patients. So a total of 6 patients in the initial phase of the study. And what it's a dose ascending dose study. So we initially give up what is the middle dose at this time, and then we'll be giving a higher dose in the second cohort of patients. And what we do in terms of the design is we inject one patient, we wait for 21 days to 28 days, depending on what is defined. And then based on that, if we do not see any signals of safety in that particular patient, the first patient or sentinel patient, then we can dose the second and third patient. Once we finish the cohort, we'll wait for another 21 days, and then we'll get to the next set of patients after conferring with the DSMC membership. So the Data Safety Management Committee obviously has to look at the data, and we have to provide them all the data in terms of safety, and then they come back and give us the feedback that we can move on to the next stage of the study. And so once we finish the 6 patients, we'll obviously evaluate these patients. The idea is, initial goal is to look at the data for the first 12 months, and then obviously there's long-term follow-up time frame, so we are going to be following up these patients for a total of 5 years. So in terms of outcomes of this study, once we follow these patients for 1 year, we look at the data, both safety as well as efficacy data at that point of time. Once we see enough data for us to get convinced that we can move on to a larger study, we'll obviously be looking at that and designing that and moving forwards. When I mentioned that patients will be followed up for 5 years total, that's from an FDA perspective. So any patient who gets into a genetic study, as you know, has to be followed up for a long-term phase. So we are going to do that for the 5 years total. But once we have enough data to convince ourselves and obviously the scientific membership that this is good data to proceed with the next stage of the study, we'll be starting that study soon after that. So that's the plan at this time. In regards to the sites, we currently have 4 sites. So we have 2 sites in California. One is in Irvine itself, which is where our Vision Research Center is also located. So University of Irvine, California, with Dr. Barry Cooperman, who's the chair of the department, and he's the PI. We have Dr. David Boyle in Beverly Hills from the Retina and Vitreous Associates. We have Dr. Kenneth Fan in Houston, who's part of the Retina Consultants of America. And then we have Dr. Jose Sahel, obviously a pioneer in optogenetics, and he's at Pittsburgh. So we have these 4 sites. One of the unique things that we do is we have patients be looked at from the efficacy side of things. We have the patients come to our Irvine Center. So safety, all the screening, all those elements are taken care of the sites, but any of the other efficacy aspects are being taken care of one site, our site in Irvine, which is firewall from us. So we don't really intervene there, but we, obviously, the reason why we started that site was basically to make sure that there's enough time, enough patience in people who test these patients to be able to test their efficacy aspects, with over time, so to speak. And the reason why I say that is because when you go to sites, the clinical sites, there are people who can do all this testing, but sometimes they're pressured for time. And so what we do is we have the patients come to Irvine. They can spend a day or two getting the efficacy testing done there. First day, if there is an issue, then they can wait over and get the complete efficacy testing done the second day. So that's an advantage. So we have the patients flying. So we fly the patients in. They can stay and get the treatment done there. The efficacy testing done there, and then they can fly back. So that's an infrequent kind of it's not the patient has to fly in every week or so. So there are certain aspects that they have to follow as part of the protocol. So the website is raytherapeutics.com. So we will obviously, patients can go on that and be able to check on the information about the clinical trial. And hopefully by the next day, we will have some data to present and be able to talk some more about it, about the study.